Welcome to 3-minute NEET PG preparation with Dr. Murali Bharadwaj on the Incus Learning app. We will discuss about the purines and pyrimidines. You should all know that the guanine cytosine bonds, they have 3 hydrogen bonds. They are stronger than adenine thymine bonds, which have only 2 hydrogen bonds. That is the reason whenever guanine cytosine is rich, then it will increase the melting temperature. So what is melting temperature? It is the temperature required to break half the double bonds, the DNA bonds, is called melting temperature. The more GC bonds, the greater will be the melting temperature, is what you should remember. Then what is Chargaff's rule? Guanine is equal to cytosine. Total amount of adenine is equal to thymine. That is what Chargaff's rule. Then purine versus pyrimidine. Remember, purine has 2 rings. Pyrimidine has 1 ring. See, easy to remember. Pyramid. How will the pyramid will be? Pyramid is only 1. Right? So, pyrimidine is 1 ring. Purine is 2 rings. Adenine, guanine are purines. Cytosine, thymine are pyrimidines. Thymine is there only in DNA. Uracil is there only in RNA, as all of you know. Then xanthine, hypoxanthine, uric acid, they are all basically purines with 2 rings, is what you should remember. Then finally, how does allopyrinol or 6-mercaptopurine function? They both are purine analogs. They inhibit the phosphoribosyl pyrophosphate amidotransferase, PRPP amidotransferase. And that is how they are the ones which prevent the purine degradation into uric acid, is what you should basically remember. So these are some of the very important points. Don't forget to subscribe to the 2000 HD videos in the Incus learning courses. Please go to